the second step, which is the melanosome development. Once the genes induced by the MITF are translated into protein, this will produce it in a formation and maturation of the melanosomes. Melanosomes typically follow four maturation stages according to their structure and the quantity and arrangement of the melanin that is produced inside. In the early melanosomes, in the stage 1 and 2, it's an internal fibrillar matrix is formed and produced, and the melanin synthesizing enzymes are also incorporated. In late melanosomes, in stage 3 and 4, the melanin starts to be synthesized and deposited in these matrix fibrils, resulting in some vesicles that are filled with this pigment. Then the activity of the melanin synthesizing enzymes is lost, and the melanosomes can be transported. Apart from the enzymes, the melanosomes contain structural fibrillar proteins that are essential for the formation and maturation. We can highlight, for example, the PML17, that is a very important component in the melanosomes and play a role in the production of internal fibril. On the other side, the melan A, that is also known MIRT1, it's a melanosome-specific protein that is placed in the melanosome membrane, which is rich in early melanosomes, suggesting that it plays a role in the biogenesis. So melan A and PML17 forms a complex that controls also the function of the melanosome formation and maturation. So the first step is the production of the melanin inside the melanosomes. There are two chemically distinct kinds of melanin, the black-brown, that is called eumelanin, and the yellow-reddish, that is called pheomelanin. The skins contain a mixture of both, and the ratio determines the visible pigmentation. The eumelanin is the major type in individuals with dark skin and hair, while the pheomelanin is abundant in individuals with red hair and fair skin. There are different enzymes that are absolutely required for the melanin synthesis in the melanosomes. Probably the most well-known is the tyrosinase that will transform the tyrosine and the dopaquinone, and it's the key enzyme in the first two steps of the melanin synthesis. Then, in addition to the tyrosinase, we have also the tyrosinase-related protein 1 and tyrosinase-related protein 2, this TRP1 and TRP2, that will be present in the transformation of the dopachrome to eumelanin. Okay. So this is one of the traditional methods we have to control the melanin, this the inhibition of the tyrosine. But this is not enough. As you can see, the melanin production is a very complex, and we need to act in different in all the different steps of the process just to have a high and a more efficient melanin control. So finally, the fourth is the transport and transfer of the melanin. So once we have the melanin inside the melanosome, this is transported along the melanocyte dendrites, okay, these channels, and then it is transported thanks to the movement of the cytoskeleton components. Once the melanosomes are in the tip of this melanosome dendrites, it is transferred to the keratinocytes by phagocytosis. This phagocytosis process can be controlled by a protein called DKK1. We know that this KK1 is involved in the transfer and it inhibits the phagocytosis of the melanin-containing vesicles by the keratinocytes. So if we want to reduce this transfer, we can increase the concentration of the DKK1 because it will inhibit the phagocytosis of the melanin, decreasing the amount of melanin that will be transferred to the keratinocytes. So in normal conditions, this process is done correctly, but as you know, the cells are constantly exposed to UV that in fact is the main responsible for the production of the melanin. But this UV, in addition to the production of the melanin, can produce some DNA damage. So this DNA damage can alter some cell function, leading to changes in the pigmentation. So even if we have some internal DNA repair, that pathways that will be taking care of our DNA, in some cases, the damage produced will produce this hyperpigmentation. So in our continuous search for new cosmetic ingredients, we found microorganisms in Tenerife, that it's in the Canary Archipelago in Spain. You can see here the map. It's an archipelago of island. And here the climate is very constant with not very high changes in the temperature, but with many sun hours and very little rain. So in these conditions, we have discovered microorganisms in the seawater, and we have extracted a plankton extract from these microorganisms. So we analyze different properties of this extract, like, for example, the expression of the melanogenic genes, the melanin production, also the phagocytosis modulation, and finally the melanogenesis in reconstructed epidermis. And finally, at the end, we have demonstrated the efficacy in an in vivo test. So let's start now with the different in vitro tests that we have done to analyze the efficacy of this plankton extract. The first test was the modulation of expression of melanogenic genes. So what we did was to take some human epidermal melanocytes, cell culture, and we add 10 micrograms per milliliter of plankton extract during 13 days, repeating the treatment in the days 3, 6, 8, and 10. And finally, through microarray, we analyzed the different genes that were expressed in these melanocytes after the incubation with the plankton extract. And then we observed some interesting results. For example, there were some genes that were down expressed. So the melanocytes express less genes than the usual. And some of them were directly related with the melanocyte activation. In that case, we can see here the endothelin receptor B, that you remember is one of the receptors that can interact with the endothelin 1, that has been down-regulated. And the same with the free cell receptor that also are directly related with the melanocyte activation. On the other side, we have observed that a downregulation of the PMEL17 and MELANE A, that if you remember, were directly involved with the melanosomes development. And also we have seen a decrease in the expression of tyrosine and tyrosine receptor proteins 1 and 2 that are directly related with the melanin production. On the other side, we have seen an upregulation of the DKK1 that, if you remember, it's directly related with the phagocytosis. So if we increase DKK1, the transfer from melanosomes to keratinocytes will be decreased. So with only one ingredient, we are affecting all the different steps in the melanogenic process. So in the next experiment, we observe how the incubation of plankton extract can have a direct impact in the production of melanin. In that case, we take also human melanocytes and we incubate it with different benchmark brightness. Like, for example, hydroquinone, we have also arbutin, we have kojic acid, etc. The control, it's the amount of melanin that is naturally produced by the melanocytes, and it has been normalized to 100%. And you can see, for example, hydroquinone here that reduces the concentration in 32.4. And here you can see the plankton extract that is in line with some of the more used whitening or brightening standard agents, like, for example, kojic acid, magnesium ascorbyl phosphate, etc. But as you know, some of these brightening ingredients can have big problems, especially in terms of cytotoxics. For example, hydroquinone, kojic acid have some toxicity, but also some problems in the stability. Some of them can produce brownishes in the formulation. So at the end, it's a double problem, so toxicity and stability. And with this plankton extract, we will avoid all this problem because it's a completely safe ingredient and it's not producing any problem in terms of stability. The third experiment is the modulation of the phagocytosis in the keratinocytes. So in that case, we produced two different kit sets of experiments. In the first one, we take some keratinocytes that were incubated in a standard cell culture medium. And we perform a phagocytosis assay. Basically, this phagocytosis assay, we have some microspheres with a fluorescent agent inside, and we want to see if the keratinocytes are able to phagocytize and internalize these fluorescent microspheres inside the keratinocytes. Okay, so this is the positive control. We have also a second experiment that is adding some DKK1 in the cell culture, in the keratinocytes. As we have commented previously, this protein is reducing the transfer of the phagocytosis. So in theory, adding this DKK1, we should observe less phagocytosis. The amount of internal fluorescence microspheres inside the keratinocytes will be lower than in normal conditions. The second set of experiments was done in combination with melanocytes and keratinocytes, and also with our plankton extract. So the third experiment is we took some melanocytes that is producing a standard amount of DKK1, the normal amount. And once we have this, we took the melanocyte supernatant with the DKK1 produced by the melanocytes, and then we perform again the phagocytosis assays in the keratinocytes. And the fourth experiment is we put the melanocytes in contact with the plankton extract. So plankton extract, we know that increases the amount of DKK1. So in theory, it will increase the amount of DKK1, so will reduce the phagocytosis. And this is what we see here. We will do some zoom here. As you can see, there are some red particles in the first picture. So these red particles are the fluorescence microspheres that has been absorbed in the keratinocytes. Okay, when we add the DKK1, the amount of this red particle is decreased. Okay, so we demonstrate that the DKK1 reduces the phagocytosis. In presence of melanocytes supernatant, we can see also some red particles. But when we add the plankton extract, we increase the amount of DKK1 and the amount of red particles absorbed are also decreased. And this can be also observed in the graph. Finally, we have in vitro results for the reconstructed epidermis. In normal conditions, we can see some amount of melanin that is produced here and transported directly to the basal layer of the keratinocytes. When we add WNT1, that it increases the melanin synthesis, we can observe how melanin is increased. We can observe this in the graph, but also in the cell culture image. You can see clearly the amount of the melanin that's increased respect to the first. When we add this WNT plus a plankton extract, we observe a decrease in the melanin synthesis that can be clearly observed in the picture, but also in the graph. We tested two different concentrations, 100 picograms per milliliter and 200 picograms per milliliter, and in both cases, we observed a decrease in the melanin synthesis. So this is in vitro. What happens in vivo? So what we did was to test a cream containing 2% of the solution of this plankton extract and a placebo cream twice a day during 56 days in a group of 24 African female volunteers with skin prototypes between 5 and 6. And then we analyzed the color with colorimetric measurements, photograph, and also some self-assessment. First, we can see here the colorimetric measurements. We analyze the luminance L and the individual topological angle. So if we want to have a higher luminosity, what we want to observe is higher luminance, but also an increase in the individual typology angle. You can see here what this individual typology angle represents. So the higher the angle, the less intense the color. So in gray, we can see the placebo after 28 and 56 days. And we can observe how in purple, our solution with 2% cream contain 2% plankton extract increases significantly the luminance after 28 and after 56 days. And the same with the individual typologic angle with a significant increase after 28 days. We can observe here some pictures, some photographs of some volunteer time 0, 28 days, and 56 days. So because sometimes it's difficult to observe the results here, what we have is analyzed this using a software of luminosity that generates a color maps depending on the luminance. So when we have low luminance, the color will be red to orange. And when we increase the luminance, the color will change to yellow and to green. And this is what we want to observe. We want to observe changes from red to green. So you can see here in 8 days, and there are some areas that, for example, in the front, the redness is reduced, and in that part we can observe more green and yellow, also the same in the nose. And this is even more easy to see after 56 days. We can see even observe some blues in the cheek that is demonstrating the higher luminance of these pictures. We have the same here. In that case, we have also some dark spots at time 0, and we can observe how after 28 and 56 days, these dark spots have been decreased a lot. Okay, and also in general, we can observe that the skin is more homogeneous and it has a more even tone. In the self-evaluation of the volunteers, 22% of them said that it helps to lighten up the skin tone. 80% said that it improved the skin luminosity, and 88% said that the skin tone appears more even. So as a conclusion, we have this ingredient that controls the melanin hyperpigmentation, just acting in the 4 different steps of the melanogenesis synthesis. And we have demonstrated that just only 2% we have good results in terms of luminance and in increasing the individual typological angle. And that's all. This is my presentation. Thank you. I don't know if you have any question.
Interesting. We always welcome efficacy studies and data on African skin. So just a question posed to you. You say here that there were 24 African female volunteers. Can you elaborate on, is it South African, Congolese? Is it just South African, Nigerian?
I think it's South Africa, yes.
Okay, so just South Africa.
But I should check. I am not 100% sure about that. But
so yeah, so it's always interesting because, you know, the different countries with the African skin have different results. So that's why I was asking, because sometimes the collation of the results, if there's 6 South African women and 3 Nigerian and 4 Congolese, there are quite different variations between when we test these efficacy, the efficacy of these skin lightening actives on the African skin. And did we see if there was an inky?
Yes.
Is there an inky of the product?
Yes, it's plankton extract.
Plankton extract.
Plankton extract, yes.
So I'm believing that these marine extracts are, I think they float around in the sea. They're having amazing results because we are seeing more and more in the industry with regards to a lot of the marine biotechnology coming out with regards to skin pigmentation. I would like you to go back to the slide. There was a slide that you did a comparison with alpha-arbutin hydroquinone. If you can go back to that slide, I cannot recall what the results were measuring. Was it measuring which activity, or if you can just find
it was
that?
Directly the amount of melanin directly analyze it.
So can you please bring up that slide?
Yeah.
There we go.
Yeah.
Okay, so our understanding is that the plankton extract has a higher level of lower level of melanin compared to using something with
exactly what we are analyzing here is the remaining melanin. So in that case, you can see, for example, the most efficacy is the hydroquinone that with the 100% that is the standard amount, only 32% remains. So the decrease is higher. In terms of the concentration, something that I would like to highlight is that the concentration of the active is always the same. So it's always 1 micrograms per milliliter. Okay. So the plankton extract could be, you can see it's more efficient, for example, that kojic acid or magnesium ascorbic or the glycerinic acid, for example. So it reduces around 50% of the total melanin content. So only hydroquinone, hydroxyresortinol, or arbutin is showing higher results.
Yes, okay. Thank you for explaining that slide.
Yeah.
All right, and Liesel, from your side?
No questions from on this side. All good.
So there was one last question, Dr. Juan, before you go. You've mentioned that you've done it in the application of a cream. How would it work in a gel-based or a serum-based?
It was an emulsion.
An emulsion.
An emulsion, yes.
Okay, perfect. Thank you very much. We enjoyed your presentation. Thank you for all the data. And once again, to say thank you for sharing data on African skin. We're always welcome to see such new data. Thank you.
Thank you very much.
If I had introduced you, I would have said, "Como estás?" That's all I know, but I will say adios.
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 Muchas gracias.
Muchas gracias. All right, and we are going to go on to our tea break. So we hope that you're enjoying the conference so far. And please keep sending questions, and we'll see you on the other side of the tea break shortly. Thank you.
Some people need their fears. They think it makes life clear. No one's on earth alone, yet everyone's on their own. Someday, one day, maybe, somehow, not now, and you will find what you lose once you have peace of mind. Most start out with great ambition. Years gone by, lose your time, lose your mind. Drink that wine, drink the wine. Look at me, pure. Everything that I am, I adore. It's the reason life is for. It's my heart that I need to be sure. Keep it pure. Be pure. Anything that you dare and more. Love the nap on my hair. I'm pure. What you love at the root and the core. Keep it pure. Some start out looking for love, settle up when they've looked enough, looked enough. Most people think they have time to begin life. They're so wrong, they're so wrong, they're so wrong, so wrong. What I dream of, if I dream hard enough, will become mine if I don't run out of all my time. Most people have intuition to love life, love your life, feel alive, oh, feel alive. Look at me, I'm pure. Everything that I am, I adore. It's the reason life is for. It's my heart that I need to be sure. Keep it pure. Be pure. Anything that you dare and more. Love the nap on my hair. I'm pure. What you love at the root and the core. Keep it pure.
Helen Playden from the Careco, and I'm going to showcase some of our exciting ingredients from a cross-section of our suppliers and some of our 2022 new launches. We have been experimenting in our concept laboratory on tinted mineral sunscreens using pigments, zinc oxide, and an exciting launch called Estergel, a natural polymer for thickening of oils and stabilizing of formulations. We formulated 2 SPF products containing organic UV filters with Polymer Expert's Expert Gel that improved film formation, and Estergel Green, which improved viscosity and film formation of a mineral SPF. Textures can be obtained with balm oil using natural oils and esters, together with Miyoshi Silica, Vitellis Jarchem Shea Butter, Estergel M and Green, and the addition of Sun Chemicals Sunshine Pigments provided color and pearlescence. Scott Bader's Texic range of bio-based high-performance thickeners with an average of 60% naturality, simultaneously emulsifier. Cationic PQ37 in hemisqualene is a multifunctional conditioning agent and rheology modifier. Texturizers from Sapphic Alcan natural range include traditional xanthan gum and a clear version showcasing the suspending properties of clear xanthan gum. We offer solutions for hair dyeing in the form of bleach and color. A fantastic leave-on concept called 8-in-1 for ageless hair from Givaudan contains Resistyle to moisturize, smooth, style the hair, and Darkenil to repigment grey hair. Givaudan's range includes visual enhancing unispheres, actives for soaps, wonderful fragrance oils, and a substantial portfolio of botanical and scientific active ingredients for skincare. Here is a pretty picture of Very Peri, the 2022 color of the year in a unisphere. Encapsulated hyaluronic acid for lip care, smoothing and plumping of the lips has just been launched and will be available soon. Our interesting range of packaging offers claims such as recyclable, reusable, natural, vegan, halal for skincare and makeup applications, in serum bottles, airless jars and pumps. We provide nail polish from suspension bases to coloring solutions. Our bases claim long wear, breathable, natural and supernatural applications. We have a spectrum of color and ranges with special effects. A wide variety of treatments for all nail problems is on offer. Whether you want to make nail polish or buy it in bulk, we can assist. Our team is highly skilled, passionate, energetic and caring. We look forward to being of service to you.
Simrise. Always inspiring more.
Leading beyond chemistry.